Microorganisms are present everywhere around us. While many are harmless and even beneficial, and some have the potential to cause serious diseases in humans, animals, and plants, scientists and researchers study these microorganisms to understand their biology, develop diagnostic tools, produce vaccines, and discover effective treatments. However, working with infectious agents carries inherent risk. To ensure the safety of laboratory personnel, the community, and the environment, laboratories are designed with different level of containment known as biosafety levels: BSL-1, BSL-2, BSL-3, and BSL-4. Each biosafety level represents a specific combination of laboratory practices, safety equipment, and facility design based on the risks of disease posed by the microorganisms being handled. As the risk associated with the pathogen increases, the containment measure become more stringent. From basic teaching laboratories handling non-pathogenic organisms to highly specialized facilities working with deadly viruses such as Ebola, biosafety levels provide a systematic framework for safe scientific research. In today's lecture, we will explore the four biosafety levels, understand their unique characteristics, compare their containment requirements, and examine how these laboratories play a crucial role in advancing science while protecting public health and environment. The goal of biosafety is not to prevent scientific discovery, but to ensure that discovery occurs safely and with responsibility. Biosafety levels BSL-1 to BSL-4. So let's understand the hierarchy of the laboratory safety. And before we focus on BSL-3 laboratories, it's important for us to understand that not all microorganisms pose the same level of risk. Some microbes are harmless and are routinely used in teaching laboratories, while others can cause severe life threatening diseases. To ensure that scientists can study these organisms safely, laboratories are categorized into four biosafety levels: biosafety level 1, biosafety level 2, biosafety level 3, and BSL-4. These levels forms a hierarchy of containment in which safety measures become progressively more stringent as the risk associated with the microorganism increases. At the foundation of this hierarchy is BSL-1, the lowest level of containment. BSL-1 laboratories are designed for with microorganisms that are not known to cause disease in healthy adults. Examples include organisms such as Bacillus subtilis and Lactobacillus species. These laboratories are commonly used in schools, colleges, and teaching institutions where students learn basic microbiological techniques. Standard laboratory practices such as hand washing, surface disinfection, and proper waste disposal are generally sufficient for safe operation. Personnel usually wear laboratory coats, and no special containment equipment or air handling systems are required. BSL laboratories emphasize fundamental laboratory skills and serve as a starting point for microbiological training. The next level is BSL-2, which is intended for microorganisms capable of causing mild to moderate disease in humans. Organisms such as a Salmonella species, Staphylococcus aureus, and hepatitis B virus fall into this category. Unlike BSL-1 laboratories, BSL-2 facilities require restricted access during experiments and additional precautions to prevent accidental exposure. Laboratory personnel wear gloves, lab coats, and eye protection, and procedures that may generate infectious aerosols are often performed inside a biosafety cabinet. These laboratories are commonly used in diagnostic laboratories, hospitals, and research institutions where human pathogens are routinely handled. The primary focus of BSL-2 is to prevent accidental infection through contact, ingestion, or exposure to contaminated materials. As we move higher in biosafety hierarchy, we reach BSL-3, a level designed for pathogens that can cause serious or potentially fatal diseases and may be transmitted through air. Examples include Mycobacterium tuberculosis, SARS-associated coronavirus, and a highly pathogenic influenza virus such as H5N1, because inhalation of airborne microorganism possesses a significant risk. BSL-3 laboratories incorporate advanced engineering controls and containment measures. Access is strictly limited to trained personnel, and all procedures involving infectious materials are performed within biosafety cabinets. The laboratory operates under negative air pressure, ensuring that air flows into the facility rather than out of it, thereby preventing the escape of pathogen. Exhaust air passes through HEPA filters before being released into the environment. Personal wear specialized protective equipment, including respirators, gowns, gloves, and eye protection. BSL-3 laboratories represent a critical bridge between routine clinical laboratories and the highest level of biological containment. At the top of the safety hierarchy is BSL-4, the highest and the most stringent level of containment. These laboratories are reserved for work with extremely dangerous and exotic pathogens that cause severe or often less. So, at the top of the biosafety hierarchy is BSL-4, which is the highest and the most stringent level of containment. These laboratories are reserved for work with extremely dangerous and exotic pathogens that cause severe or often fatal diseases, and for which effective treatments or vaccines may not exist. For example, Ebola virus, Marburg virus, and Nipah virus. BSL-4 facilities are often physically isolated from other buildings and equipped with highly sophisticated containment systems. Scientists work with fully enclosed positive pressure suits supplied with dedicated breathing air. Entry and exit procedures involve multiple layers of security, airlocks, and extensive decontamination processes. Every aspect of laboratory is designed to ensure that this deadly pathogens remain completely contained. BSL-4 laboratories are rare and represent the highest level of biosafety infrastructure and available in the world. When viewed together, these four biosafety levels illustrate a progressive increase in containment and protection. BSL-1 focuses on basic laboratory safety for harmless microorganisms. BSL-2 introduces precaution for moderate risk pathogens. BSL-3 provides enhanced containment for serious airborne infectious agents. And BSL-4 delivers maximum security for the world's most dangerous pathogens. Understanding this hierarchy helps us appreciate why different microorganisms require different level of containment and highlights the special importance of BSL-3 laboratories positioned between routine diagnostic facilities and maximum containment laboratories. BSL-3 facilities play a vital role in studying pathogens that pose significant public health threats while ensuring the safety of laboratory personnel, the community, and the environment. So, the simple way to remember the hierarchy is BSL-1, which learns safely harmless microorganism. BSL-2 work carefully moderate risk pathogen. BSL-3 contains strictly serious airborne pathogen, and BSL-4 maximum security deadly exotic pathogens are involved. So we work with those pathogen in BSL-4 laboratories basically. This progression from BSL-1 to BSL-4 reflects the fundamental principle of biosafety: the greater biological risk, the greater level of containment required.